Hi, my name is Courtney Collins, and I am a postdoc at CU Boulder with Drs. Katie Suding and Sarah Elmendorf. And today I will be discussing my research on how open-top chamber warming experiments consistently influence tundra plant phenology across large spatial and temporal scales. So as you likely know, tundra ecosystems are changing. They are warming at about 2 times the global average. And this is creating dramatic shifts in the structure and function of these ecosystems. And changes in plant phenology are a key indicator of how these ecosystems are responding to climate change. We can see this in the figure by Piao et al. 2007, where we see large shifts in the global trends of green up or leaf out, primarily to earlier green up, while we have mixed patterns of responses to warming for dormancy. We have both later and earlier dormancy, shown in the green and red, respectively. And this is leading to, again, mixed patterns in growing season length, where the growing season is both shortening and lengthening in response to global climate. And this is creating large consequences for plant pollinator interactions, herbivory, and above and below ground carbon stocks, just to name a few. So in order to better understand these patterns and get a mechanistic understanding of what is happening, we need to have experimental approaches. And warming is the most consistently predicted global change impact to the tundra. So open-top chambers are a widely used passive warming experimental approach to study this response of plant phenology. And as we can see here in the figure by Healy, they create a nice degree of warming shown in this thermal imagery, about 1 to 3 degrees above ambient inside of these open-top plexiglass chambers. However, the understanding we have of how plants respond to experimental warming is varied. In 1999, Stephanie Arft wrote a synthesis looking at open-top chamber warming across the tundra and found that ecosystem type, plant functional type, and years of warming influenced how plants respond to experimental warming. In 2012, Lizzie Wolkovich found that experimental warming actually underestimates the effects of climate warming on plant flowering and leaf out by a significant degree. And then most recently in 2019, Janet Privé actually found that warming is advancing flowering for these later flowering species rather than earlier flowering species. And this is shortening the length of the flowering season. But that experimental and observational warming or ambient warming are predicting similar shifts in these flowering season lengths. And so we think that there's still a lot to know about how plants are responding to these experimental treatments, and we've defined some critically unresolved questions that we are trying to answer with this research. Primarily, we want to know, does experimental warming differentially affect reproductive and vegetative phenology? As a lot of these experiments either look at one or the other. We also want to know whether they shorten, lengthen, or have no effect on the duration of these phenoperiods, including the growing season length, the flowering length, and the fruiting period length. We also want to know if plant responses to experimental warming are variable across spatial and temporal gradients in both resource availability and ambient climate, and whether plants can sustain their responses to experimental warming over very long time periods, or whether these have just short-term effects and then thermal acclimation kicks in. So in order to test these questions, we've been tapping into the International Tundra Experiment, which is a long-term project of monitoring Arctic and alpine tundra plant phenology and their responses to climate change. And this has been going on since the early 1990s at many of these sites. And they have both ambient observations to climate warming, but also experimental treatments, primarily passive warming chambers, these open-top chambers. So this is a map showing 18 ITEX sites that we've included in this experiment, both in the alpine and Arctic tundra across the globe. So again, like I mentioned, we've been using the ITEX publicly available data set, but we have created a recently updated version with observations all the way to 2019, spanning the last 30 years approximately. And we have 18 sites and 46 experimental locations in this data set of over 100 plant species. So really broad responses. And we've also been able to include 6 plant phenophases from across the entire growing season. So green up, start of flowering, end of flowering, fruiting, seed dispersal, and leaf senescence, to try to get a really complete picture of how plants are responding across their entire kind of life cycle in the tundra. So to try to understand these responses, we have used Bayesian hierarchical modeling, and we've been able to predict these observed phenology data in our large data sets where the response is the day of year of a phenology event occurring. And we use interval censored hierarchical models to help account for this uneven sampling frequency across different sites. So every day of year response is given both an upper and lower bounds. And the upper bound would be the first day which this phenophase was observed, whereas the lower bound would be the closest preceding survey date that a plant was observed not to be in this phenophase. And so then the actual estimated day of year is somewhere between these upper and lower bound dates. And we use fixed effects of treatment, so treatment being either open top chamber or control conditions. And we're also interested in treatment interactions with several spatio-temporal factors, which I'll talk about in the next slide. And to account for these large differences in species, site, years, we've included random slopes of treatment by species, site year, and site by subsite. So again, for these model parameters, we have treatment, which is just a categorical predictor of either open top chamber or not, so control conditions. We're also interested in interactions between open top chambers and these other different parameters here. So first we wanted to know whether the effects of experimental warming are interacting with the years of warming, so how long the duration of this experiment has been going on at a particular site, or the latitude of the particular site, whether or not the warming chambers are more or less effective at high or low latitudes, as some work has shown that they may be more effective in really cold, high latitude sites. We're also interested in the interaction with soil moisture, as some work has shown that open top chambers will warm less in very wet or moist sites due to buffering of the water in the soil. We're also interested in how OTC deployment time period, so whether it's year-round and they are left on throughout the winter or just placed on in the summer. As you can see here, this is a year-round chamber, which at the end of the winter has really created kind of a snow fence and accumulated snow on one side, but then within the chamber, we actually have pretty little snow because the plexiglass is allowing reflection of the solar radiation and melting out snow rather quickly. And finally, we were curious about the interaction between ambient climate and open top chamber warming. So we calculated site mean temperature averages during species phenology windows. So the average day of year that a species had a phenology event and the preceding 30 days prior, we averaged those daily climate data to calculate this site mean temperature. And then we also looked at this across years, how different was each year from that average site mean of a species phenology window. And so getting into some of our results here, on the x-axis, we have the number of days difference in the phenology response of plants that were growing in open top chambers versus plants growing in control conditions. And so these are density plots, and they show the distribution of the modeled parameter estimates for each phenophase. And the zero line here would suggest no change, and anything to the left or the right of this zero line would suggest a difference between open top chambers and controls. And then the red line and the gray line are the 90 and 95% Bayesian credible intervals on these estimates, respectively. And so overall, what we can see is that we had an advance in green up as well as flowering, end of flowering, and seed dispersal. And we had a delay in leaf senescence. There was no change in fruiting period as our estimates and credible intervals crossed the 0 line pretty significantly. Interestingly, we also see that reproductive phases shifted much more than vegetative phases. So these reproductive phases shifted on average between 2 and 2.8 days, while the vegetative phases shifted only about 0.6 to 0.8 days. We also see that green up and leaf senescence shifted in opposite directions. So green up was advanced while leaf senescence was delayed. And this would suggest a potential lengthening of the total growing season by about 1.4 days. In general, the interactive effects that we tested between treatment and spatiotemporal factors were very minor. We only found about 4 significant interactions out of 42 that we tested. And so to us, this suggests that plant responses to experimental warming are largely consistent across all of these different species, functional groups, and geographic regions, and that responses to experimental warming can be assessed both in the short term and over very long timeframes. But out of interest, I will share with you the 4 significant interactions that we did find. We saw that soil moisture influenced the flowering response to open-top chambers. In that flowering at dry sites, which is shown in the red here, was about 1.3 days earlier on average than flowering at moist sites in open-top chambers. And so now we have our difference in days on the y-axis, and the 0 line is this dotted gray line here. So anything above or below the 0 line would suggest a difference in response between warming and control experiments. And so we can see here in the box plots that dry subsites, shown in the red, in general flowered earlier in open-top chambers. This was earlier than our moist sites. There was no difference between the dry and wet sites, but we did have lower replication at our wet sites. And so this suggests that tundra plants may be responding non-linearly to the combination of thermal and moisture stress when they're flowering. We also saw that seed dispersal varied by the deployment period of these OTCs, where seed dispersal was significantly earlier in OTCs that were left on year-round than those were in the summer only, and this was about 5 days earlier. And so this might again be due to what I mentioned previously, that there's earlier snowmelt and likely warmer thermal accumulation in these year-round OTCs, which are left on throughout the entire winter. And this might be advancing this entire growing and reproductive period. We also saw that green up responses shift over the duration of warming, and that they in general were dampened with increasing years of consecutive warming. So our overall pattern in green up was slightly earlier shift, which we do see here in about the first 10 years or so of consecutive warming, but that between 10 and 20 years, this effect becomes very minor. And then after 20 years, we actually see a slight delay in green up. So this suggests that plants may be having some thermal acclimation over time to these open top chambers. We also saw that seed dispersal in our open top chambers varied by the site temperature during these species climate windows. So species with colder climate windows during their dispersal period had no advancement or even a slight delay in seed dispersal, whereas the average overall was an advancement in dispersal. This was primarily driven by these warmer sites here that had species with much warmer dispersal climate windows. And interestingly, seed dispersal was the only phenophase with sensitivity to ambient climate under experimental warming. But seed dispersal is often not included in a lot of these larger syntheses. So we really think that having a broad range of phenophases is really important to capture the full response of plants to potential climate warming. So in conclusion, we saw that there were stronger shifts in reproductive versus vegetative phenophases in response to experimental warming, and that this is leading to a likely lengthening of the growing season, but no change in the flowering or fruiting periods. There was little variation in our treatment effects over space, time, and ambient climate, and plant responses to open-top chamber warming are largely consistent across species, functional groups, and geographic regions. And so with that, I'd like to thank so many people that helped me with this work, including the scientists and the iTex network for all of their collection of this amazing phenology data. The photo here of Anne Bjorkman collecting some data over on Alexandra Fjord and Ellesmere Island in Canada, and my Suding lab members at CU Boulder, and funding from the Institute of Arctic and Alpine Research at CU Boulder as well. Thank you.